So with the help of this diagram, we are trying to understand how a protein which is being synthesized is brought inside the endoplasmic reticulum. So we have discussed about the SRP protein. So these are the, this is the SRP protein, the green color SRP protein, a cytosolic protein. The SRP protein has got two properties. One property, the SRP can bind to the signal. Where is the signal present? In the new protein which is being synthesized. It can bind to the signal. So which means the SRP protein should be able to bind to the signal which is green in color. And the second property of SRP protein is it can bind to its receptor which is located, so the receptor which is located on the endoplasmic reticulum. Now assume that this is the, this is the endoplasmic reticulum membrane. This is the endoplasmic reticulum membrane. This is the cytosolic side. This is the lumen. Now, if the receptor is a dimer, it has an alpha chain and it has a beta chain. The alpha chain is fully on the cytosolic side and the beta chain is binding to alpha chain but embedded in the membrane. So we have shown here, this is the beta chain which is there in the membrane and this is the alpha chain. So this is the basic structure and these are the two properties of the SRP protein. Let us unfold how the protein is brought inside. So as we can see, this is the red one is an mRNA molecule. The ribosomes will bind on it and the protein is being synthesized. Now the green one is the signal. Signal is released first. As the signal is released first, we have seen about 70 amino acids signal along with the first the first few amino acids. Once the signal is released, the SRP protein comes into picture in the cytosolic side. So the SRP protein binds. So the SRP, we'll use the green, the SRP binds here and it will take up the signal inside, recognizes the signal. Where is SRP binding? In the P54, that methionine region which we showed. There it binds, it identifies. Once the identification happens, now there is an interaction happening between the ribosome, SRP, this new protein with the endoplasmic reticulum. Now that is a figure number 3. In the endoplasmic reticulum, it will come to the receptor. It will form an association with the receptor. It will form, it will identify. Now what is interesting? Along with this receptor, there is another protein. We will look at this protein in a short while. This protein is a translocon. Translocon. A protein which will allow another protein molecule to pass through. This translocon has a channel here. Now this channel will always remain closed. Because if it remains open, other molecules, ions, etc. will pass through from cytoplasm to the lumen. These two are maintained in separate, distinct places. So therefore, this channel will always remain closed. How to open this channel now? This can be opened. This ribosome, the 60S ribosome binds exactly on top of it. Okay, the 60S ribosome binds exactly on top of it and then it will insert this signal with the help of the SRP protein. It will insert the signal through this channel and it is allowed to pass through. The signal is allowed to pass through and in order to carry out all these, two GTP molecules are required. 2 GTP, where are these 2 GTP required? One GTP will bind to the receptor, another GTP will bind to the SRP protein. And 2 GTP molecules are required. These 2 GTP will be cleaved into 2GDP plus 2PI, that energy is utilized for pushing, translocating the protein inside the lumen of the endoplasmic reticulum. Meanwhile, what will happen? The SRP gets detached, and this SRP can be used again and again in order to identify a newly synthesized protein, a secretory protein, and it is identified, brought into the translocon, and then it is transferred. This is the mechanism by which a protein is translocated inside. Now the translocated protein, once the translocated protein, okay, so let me put it here. The protein is, the protein is being translocated. Now the translocated protein will have its signal. Now this signal is cleaved by this protein, and that protein is a signal peptidase. Signal peptidase will cleave, remove the signal, and now this protein can be properly folded. In the beginning, we have seen that the complicated mechanism involves three things. One is its identification, second is a translocation, then is the folding. So now we have brought the protein inside the lumen, now it is folded, it is a functional protein, it can be secreted elsewhere, or this protein can be transported to another place, or it can carry out the function in the lumen itself. Now if the protein has to be embedded in the membrane, it will have something called a stop signal. In another video, we will elaborate on this, how these stop signals will help in inserting a protein in the membrane, especially the transmembrane protein. Before we wind up this session, we need to look at this translocon. Now, translocon was identified by chance. They were looking at the role of a translocon, especially in the yeast. So what they have found in the translocon is they mutated the translocon. This translocon in yeast is called Sec61. It's a protein. In mammalian system, the same Sec is there. It has got three forms: Sec61 alpha, beta, and gamma. Many of these translocons get assembled. The Sec proteins get assembled to the translocon which we have seen. Now this translocon will have a kind of a cylindrical tubing. Okay, this is the protein, and it will have a kind of a cylindrical tubing with about 8.5 nanometer diameter. So it is through this tubing the protein is allowed to pass through. It is through this tubing. A wonderful experiment was carried out in order to prove this. A beautiful experiment. Yet it is highly skilled experiment. So what was done is they took an mRNA of prolactin. Prolactin, as you know, it is a protein, it is a secretory protein with 198 amino acid. So first of all, this mRNA is modified. In the stop codon of this mRNA is removed. If the stop codon is removed, then you know what will happen? The mRNA will keep on translating. Okay, the stop codon is removed. Now this protein has a lysine residue somewhere in the middle. Now they wanted to manipulate this lysine residue. What they have done, they took either tRNA for this lysine residue. That lysine, they modified it. They modified the lysine residue. So they modified with a cross-linking chemical. A cross-linking substance is added to that lysine. So therefore you know that if this lysine is inserted into the prolactin, it will have the cross-linking substance present. And this cross-linking substance, under illumination, it will react. Under heavy illumination, it will react, and it has a tendency to go and bind to the protein which is closest. So, this is the background. Now let us see what has happened. Assume that this is our translocon or SEC61 protein. It is forming a complex. What was observed is in the ribosome 60S, this is 60S and this is the 30S subunit. If the 60S subunit will come and sit exactly on top of it, so that if the protein which the 60S is making is not leaked into the cytoplasm or anywhere else. It is directly transported into the cylindrical structure of the translocon. So therefore, if the protein which is being made, it is allowed to pass through. And the protein will keep on making. Why it will keep on making? Because if this is the mRNA, this mRNA will have either tRNA, okay, and it will keep on making. Now there is a problem. If the problem is in between, in this prolactin protein, this is a prolactin protein, in between you have inserted a lysine residue with a cross-linking. So this is the lysine residue with a cross-linking, and that will not pass through. Somewhere in the middle it gets stuck, and it is stuck, and this protein cannot move through the translocon. It is stuck here. So no more translation can occur. So when the scientists realize that, okay, no more translation occurs, and this cross-linking lysine residue, it is stuck there, it is illumined. In the illumination, what is observed? This substance has to bind. It will force the protein to bind to the nearby protein. And it was observed that it is binding to SEC61. And now we know the basic structure of this translocon and how this protein is being translocated. All this has been revealed. Several modification of this experiment was also carried out by allowing this cross-linking lysine at multiple places in the mRNA, so that it is at various places it will be there, and then they have observed where is this protein going and binding, prolactin going and binding. So these were very, very simple experiments that were carried out, and these were the experiments they used in order to prove that in the first 30 to 40 sequences is nothing but the signal, and that is allowed to pass through first. Then you have another 30 signals, 30 amino acid, that total 70 amino acid must pass through, and then only the remaining is the protein. So these were interesting experiments that were carried out in order to prove trafficking of protein to endoplasmic reticulum. We have heavily focused on the experiments that were carried out in order to understand this mechanism, so that we get inspired to see how a simple thought and its meticulous implementation has resulted into what we know today. We are not perfect. This is not a complete story of the translocation of proteins to the endoplasmic reticulum. This is what we have focused is the co-translational translocation.